So going into this last phase, this last part of the workshop, I'm going to ask for all your patience because this is where there were the most variables that were hard to plan in advance. The purpose of this is to get a sense of the room, of overall enthusiasm for the ideas that have been raised, that came up in the topic sessions, which are a synthesis of everything that came before. It is not to definitively select these ideas. It's just to help us. When we've seen this done before, it's very useful in helping to categorize and appreciate some of the ideas, how to lump them or perhaps split them, and to generally help focus us on the most promising ideas going into the final session. Again, it is not to definitively select the ideas. It is almost 100% likely that we're going to take the products of this and think about them. You've seen how much overlap there is, or I would say reinforcement there is between each of the 4 topic sessions. There's at least one item that appears essentially on all 4 of them. We don't have time right now to reduce all those to just one list to vote on for all 4 sessions. So we're going to vote on each of the 4 sessions in turn, and use that just to stimulate the final discussion. A couple of general things. Your votes are anonymous. Everyone in the room can vote, and you will be sent the .storm URL by Chris Wetterstrand. Is that correct?
Yes, and you should have received 3 already.
Okay. So this will come in the form of so you tell me what they're going to see.
We got emails.
Okay.
So we've split each topic up into a board.
Chris,
Just repeat what I said. Each topic is a board. There'll be a message for each board.
okay, each topic will be a board, and there will be a separate message for each board.
Can you hear me? Can you hear me now?
What?
It's not turning.
Not turning red.
One of these is on. Maybe it's this one.
Oh, okay.
You're now on.
I'm on now. I believe that the NIH folks have not received an email, so
That's right.
they shouldn't not ignore it. They just actually haven't gotten it.
Yeah. So what we're trying to do is enforce this idea that if you're an NIH employee involved in planning this meeting, or you're NHGRI extramural program staff, which means you almost certainly were involved in planning this meeting, you shouldn't be voting. We'll get our licks in later.
Note that the emails don't have the topic number in the email. You just have to click on each one to do it.
And Chris, did they come in order of the topics as they were presented? Okay.
Yes.
So
we've gotten 3 of the 4 so far.
We're going to vote in the order of the topics as they were presented. What we didn't know before a little while ago is how consolidated the ideas could be. Since there are more than 30 items in total, for sure, there's probably closer to 45, maybe more. We're going to vote once on each list, as I said. Everybody is going to get 6 votes. Now, we can enforce the 6 votes for each round. What we can't enforce, but we're going to tell you to please abide by, is please give no more than 2 dots for any item. Because again, we're not trying to pick winners here. We're trying to gauge some relative enthusiasm. We also, again, understand that the items on these lists are expressed differently between lists. They're categorized slightly differently between lists. We also understand that some of these ideas have been developed in great detail, and some of them are very interesting ideas that don't have as much detail. And each of those facets will appeal to some of you more than others. So we do want you to try to vote the range.
Now, unfortunately, we can see the vote. Is there any way you can wind us to the vote? Okay.
What was
All right, and please keep in mind the following as you vote. Again, what is the most valuable thing to do to advance genomics? And is NHGRI uniquely positioned to take this on? We hope that's also a criterion. And we are fine if there are long-term ideas or short-term ideas, whatever has come up in the lists.
There are 4 boards, right?
There should be 4 boards.
Well, yeah, there are 4 boards, but each, when you click on like the first one, it's got many screens.
Right.
My main question, a couple of us are asking, how do you actually cast your vote? I did. Okay. Oh, so then that thing works? Okay.
Could we maybe show the first page of whatever board people are working on so everybody's on the same one?
It does say topic 1 in the email.
So some of us have gotten, or at least I only 3.
Yes,
Yes.
they're working on 4.
We're working on 4.
And people are still editing the final results of 4, so if you
Just
could
work on
finish
the
that
top
up,
3.
that would be great.
So,
Six dots for each topic.
Okay, so whoever is in topic 4, stop editing it?
Wait, we're not editing. We have not edited it.
Somebody is. I'm watching them edit.
Karen might be, because we gave her permission.
if you want to cast a vote, you click on the open dot, and then you'll get a little green one, which says you voted on it. You do have to put your name in at the top for that to work, is my understanding.
I'm just going to be ignorant again, I'm sure. Over here. So for each topic, how many dots we get to?
6.
6.
And only give your favorite topic if you want to.
I don't get it.
You, like, if this is like really, you want to use up 2 of your 6,
good?
you can use up 2 of your
We're
6
good?
on one topic.
So all
Oh, so
4
you get 6 across all 4 topics?
No.
6 for each topic.
Each topic has like 10 options. So
Yes.
you can make 6 choices.
Mike
Okay.
Pazin.
So on your computer screen only, your votes are green dots.
Right.
And one reason that's important to know is if you want to change one of your votes, you click on one of your green dots, it erases it. Now you can put it in a different.
But you have to count up to 6. It doesn't tell you. I understand that. Just
So
tell
it gives the option to add a card, but it says awaiting approval. So are you
Don't
guys-
add cards. Just
Yeah,
vote.
don't add cards, please. So I guess it makes more sense for everybody to go through each of the 4 topics and vote all the cards on each topic.
Yeah, which is going to take a
And
while.
when everyone's done, we'll be able to bring up the results.
And it keeps moving, so it's hard to vote.
Sorry.
Okay. I got a green card. It's actually hard to read because of the vote. Okay.
That's what it's striking. Okay.
Question, someone said there were multiple screens per topic, but I only see one.
Well, it's just you may have to scroll to see all of
yeah,
the.
okay.
So my understanding, I didn't get one, but there are what, 9 or 10 cards per
Yeah,
topic?
Something like that.
Okay, so if you don't see,
Topic 1 was very wordy, so it's lots of screens.
Put a dot here.
Oh.
Oh, you didn't sign in. Ah, okay. And now I'm signed in? Oh, I see it. Okay.
Any other questions from people?
I see the open dot moving.
Unfortunately, it jumps around as the votes go in.
There's no submit, it just stays,
Eric, what was that?
When we're all done, and we've decided how to allocate our 6 dots, do we press
There's
finalize?
no submit.
Just leave.
You just leave.
Yeah.
And our dots will be tended to?
You can see everyone else, yeah, I believe so, yeah.
Did you get the fourth? Is the fourth?
The fourth one, I'm not sure is out yet.
The
Oh,
fourth
okay.
one is out.
I seem to have gotten away with 8 dots, and I feel obliged to.
No, that's why it's an honor system. It's an honor system.
Richard? Honors. Okay, not familiar.
Oh, God. Remember, this may still be being taped, okay?
We gave you extra dots.
You never know. This could be considered the most interesting part. Uh-oh, it stopped. I lost all my dots.
Uh-oh.
1, 2, 3, 4.
Anyone else
I
have a
saw-
question?
Did anyone else just have it stop? Are you still having open? You still?
Yeah, no,
That's.
yeah.
Oh, there we go. Mine stopped. Oh, there we go. Why won't it let me vote?
Do you need help?
It's. Michael Zodi, what? Oh, all these people joined. Oh. It won't let me. Whatever. Okay.
Let me know if you need help. Mike,
My open dots disappeared.
Mike Pazin, can I ask your help for a minute?
Do you work for this company? My open docs disappeared. It won't let me vote right now.
Have you already done 6?
No, I haven't, but like you just said, it's not. I'm pretty sure I've not done 6.
Yeah, I don't know what to say other than
get out.
and see how many you have. You have 1, 2, 3,
4 or 5.
5. You should have 1.
And other people, you could, I could have voted.
Yeah, I don't know what to say.
I'll
other than
I'll click back
For
up.
now, I'll finish out your other
Yeah,
cards and then
yeah.
come back to this. I don't know if it's a refresh problem or it's the cheap one.
Well, hopefully I'm the only one who's being blocked. Maybe they suddenly decide I work for NIH. Okay.
4.
The fourth came out at 9:34 a.m. Oh, 10:34, sorry. My computer's on Central Time. Oh, okay. Two, 3, 4, 5.
Someone's going to say pencils down.
Yeah, does make sense. How is it ordering?
They may disrupt the system.
To this?
Yeah.
So can we vote on topic 4 yet?
Yes.
Yes.
Topic 4 is now available for voting.
This is unexpected.
I just don't remember how to do all the way down below the names and see if there are any options. I thought it would stay in the order I put it in. Nope.
No.
Or
One
rank
right
by
there.
age. Then you can at least have them in the order of. And most rows are now going to be at the top of the page.
3, 4,
And I guess the green is the lead.
Good, this makes sense. That's great. Thank you, Chris.
Okay. Should we give people 4 more minutes or something?
Yeah, I just want to make sure they get a chance to.
Yeah, no, I'm
Think a little-
just,
We have some time.
yeah, no, it's fine, just,
Big themes missing here, like genome assembly, function privacy. I think like those are the main ones. Training, yeah. Where's the? Oh, that's the assembly. Yeah. Okay. Yeah. I think that covers it.
Yeah.
Jump back up when it's ready to transfer to the next topic.
Can I get a quick show of hands who's still voting? Okay, we'll give it another 10 minutes.
You trying to pull out the leading ones or just thinking? Yeah.
because then,
Yeah. Yeah, this one looks like it's getting a lot of votes. Like sort of alternative. Yeah. I think that one might have been too, but let's see. I haven't looked at it in a while when I voted it.
So some of these might be concordant when we run discussions. Some
Yeah.
might be discordant.
Right. Okay. Organize it. Thanks. Super helpful.
Super helpful.
At the bottom of the screen,
We just discovered it.
we can label it. Yeah. So making sense.
Yeah. Working quite well.
Do you guys want to go through each one in turn?
Each of the topics.
Yeah. Just take a look at the votes each one in turn and just I could quickly.
Well, can you rank? Can you just
You
make
can
a rank
button at the bottom to rank.
Oh, is there a rank?
And then kind of like
summarize what top three is.
Yeah.
So we can't, can we real time take those top, let's see, there's 3 in each category and then revote?
A lot of them are redundant. There's a lot of redundancy between the topics.
Well, I see, so can we just make a list?
We can do that.
Can we just make a list? So, okay.
I don't want to revote.
Yeah,
Today.
on the, okay, got
I'm
it,
gonna
got it, got it.
take these
Some
data.
flex space, yeah.
Think about
Yeah,
collecting
I got
what
it.
we want to do
Yeah,
next
I
week.
got it. Where was the thing, Jay? Where's the, at the very bottom of the, oh. That's good. They collaborated, let's say, on that one. I don't know. Evan will be happy. So that's the top.
In different ones, yeah. Yeah, it's like number 2
very
on
close
the
there.
first sheet, but it's the way it's written and also what it's competing with. Also, the voting is sort of playing out a little bit by like what people are, how many of each types of people, because people just vote with their hands. Yeah, which
To be expected.
is to be expected. You said 10 more minutes, several minutes ago. Oh, right.
If I could get another quick show of hands of who's voting, still voting? Couple people.
I'd give them 2 minutes. This is not the final. It's just like in your college. So what's our schedule?
I could keep up a constant banter.
Well, that's why, no, we need to end it because we need to close it so people don't start changing their votes and just and export the results.
Want to do the pencil snap?
Well, no, and then we have to export the results.
Yeah.
Yeah, that's what I'm saying. We got to close the vote.
I can close the votes.
Please put away your notebooks.
Right.
Right, exactly.
All right, I think that we should get going. I know people may still be voting. I'm assuming that everybody is done with topic 1. So just a quick preview of the things at the top and the middle and the bottom.
So you're going to close it. So is there an export function?
Can we close the poll?
I can close it.
If you could, can you close them one?
Yeah, that's the question.
Okay,
We need to be able to see the
Please
vote.
close topic 1. So at the top are experimental methods development, functional genomics at scale, statistical and computational methods development.
What are you?
So at the bottom, you can either look at the screen down
Oh,
here,
rank by vote. Okay,
or you can go down to the bottom on your own screen and rank by vote.
got it. Okay.
In the middle are...
Adam?
Yes.
Is the view you're showing us now already ranked by vote?
It is already ranked by vote.
Thank you.
In the middle is interoperability and usability of data resources, perturbational combinatorial functional genomics and microscopy at scale, Mendelian disease gene identification, increased budget for training and outreach, development of model organisms and model systems to identify genetic architecture of Mendelian and complex traits and evaluate candidate variants. Here's another wide sharing of data, and some of the issues around privacy and making sure the consents are right. And then rounding this out, at the end are continued study of common complex disease, where the sequence would be coming presumably mostly from outside of NHGRI, and the rest would be done, or much of the rest would be done within.
Actually, wide sharings at the end. It's just.
Is it, does it, oh, it got.
Yours, that's so weird. Yours scrolled differently, whatever.
These are fairly.
Yeah, yours is like mine.
These are pretty close.
Yes.
It's just showing yours and where your scores are.
Okay.
But not that. That's not the summary.
Ah.
So
we don't see it.
Oh, yeah, so just so people are clear, if you hit rank by vote, no, that's not right either. It's not ranking by my vote. No, it's not.
No, not by your vote.
Ranking by
You're
total.
collected by total.
If you click rank by
see
votes
where
at the
your
bottom, it
votes are.
ranks it according to the whole
Yeah.
list. I mean, to everybody's,
Yeah.
at the very bottom. Click
all the
that.
way at the bottom on the right side.
But
Yeah, but.
I don't think you clicked it yet.
Yeah,
I have maybe something, maybe some votes were changed before, since I did it.
okay, so I
Can
do
you
think
refresh
it's
it, Adam?
important that the vote be closed, and then maybe you should refresh your screen.
I think that did it.
I did,
Okay.
but it is still appearing in a funny way.
Okay, it'll be easier for the others where there aren't as many words, so you'll be able to really see them.
In general, the top ones are at the top and the bottom ones are at the bottom, and so I think from and they're very close to each other within those, so I think it's probably fine from a Gestalt point of view.
And I don't think there's anything terribly unexpected.
But is there something we should talk?
So topic 6 is the highest ranking?
Yes.
Number 6 is the highest ranking.
It doesn't
Should we
actually
close
give
the
you
other
the
topics and review them real
Yes.
quickly and then move on
to a discussion?
Yeah, do you want to go over each topic? We can do
I
that.
think we should close them, and then you can just quickly review them.
Okay.
And then we can.
So
And then we should
everything
and then we're gonna
should be
discuss all
closed, and you
together
want to
in
go
one
on to the
block.
next topic, just to take a quick look at the results.
John, you have a question?
Sorry, I just saw the pulled up topic 1, but apparently it's closed.
Yes.
Yes.
Okay. I would just close them all.
And Chris, how do we bring up the next topic, which would be topic 3, I believe.
Oh.
Just do them in order.
This is
Two.
in the
Close
order-
two
in
and
the
then
order we
review two.
discussed them.
No, he wants to do in the order we discussed them.
I didn't lock this one yet. you go.
Okay, in predicting and characterizing functional consequences of genomic variation, at the top is complete atlas of genes and their function, complete atlas of elements under selection or cis-regulatory elements, training, way up here, high-throughput measurement of variants in organisms and organoids, high-throughput measurements of trans-regulatory effects, the 1,000 Epigenomes project, atlas of nuclear architecture towards the bottom, high-throughput approaches to accurately measure environment towards the bottom. So I think that's clear. All right, Chris, how do we go to the next one? I just go back. And then the next one is topic
4.
4.
Yeah.
We just did 3. We're doing it in the order in which they were discussed.
And then go down to the bottom to do the rank. Click on rank by vote. Then go back up.
All right. For data resources, methods, technologies, and computational capabilities, at the top is telomere-to-telomere assembly of human genomes, large-scale perturbation in pools of cells, followed by high-content, high-resolution data, build interacting data platforms, variant portals, and visualization platforms. In the middle is transferable gene regulatory or multi-scale models, resources for engineered models, mechanistic and machine learning models, and then a bunch towards the end, queryable, accessible queryable biobanks, comparative genomics and epigenomics at some considerable scale, next generation QTL study with high resolution single cell and spatial profiles and attendant data, predictive models, benchmarking data sets, and training in genome literacy for the general public. And the last one is
Topic 2.
topic 2.
Oh yeah, we need to do the rank.
And basic research questions that anticipate clinical needs. Again, up at the top, we see deep perturbation profiling, lots of molecular readouts, GTEx on steroids in the first sense, single-cell multi-omic technology, tools for data visualization, larger structural variant detection, especially in the clinical setting, computational biology workforce,
And then a whole bunch of others.
then a bunch of others at the end. Better tools for evidence, somatic variation, deep phenotyping. Okay, with heterozygous, yes, loss of function. I see what this is, yep. Genomic privacy and computational workarounds. Concentrated work on important genes in human health, environmental exposures, family, lifestyle into PRS models, and assessment of off-target effects for genome engineering. So before I turn it over to everyone for general remarks, I'll just say that it's clear that there's some things that have come up that are similar between topics that have come up and again towards the top, and then there's some interpretation for the rest. So, you guys want to reactions before?
Well, I'd like to ask if anything was really missing.
What?
Is anything missing?
Missing in the sense that topics that were not on this list and did not have a chance for airtime. Yeah. Michael?
I just wanted to point out, in case this has gone missing, that the only topic in any of these lists that involves a significant amount of high-throughput sequencing of whole human genomes was common disease, and it wound up on the bottom of the list that it's in. So does that mean that we've all agreed that over the next 10 years, NHGRI should not fund any large-scale high-throughput sequencing of human genomes? If so, that's fine, but
This is not a binding vote.
I don't want that to go missed, because if we're all just assuming that's going to happen, but we haven't given any guidance about what it should be directed towards, I think that's something that's missing.
It might depend on the electorate. So, you know, they also are going to have a meeting on genomic disease, and it's possible their meeting on genomic disease could reach a different conclusion. Right. There are probably limits to taking the temperature of any one room. I wouldn't, you know, overinterpret anything.
I agree, but I do think, Mike, I do think it's quite reasonable to remark on this, and I agree. And that whole item actually has some nuance in it, which goes beyond the headline.
But I would actually disagree. There's a lot of these topics that scored at the top that's going to make you mutate every base and then do RNA-seq on every cell, right? So those projects actually have a lot of sequencing in them. They're not just different individuals with a disease.
And the doing functional genomics in real people, you would be getting their genomes. I thought that was implicit there.
No, but that's not the 50,000.
No, that's not the 50,000. I agree about that. But
true.
Yeah, Rick?
Aviv was going to say.
Aviv.
I just want to make a small comment about interpretation, because some of these subjects actually repeat themselves across topics, but rank very differently.
Yeah.
It means, I mean, there are some things like here, the single-cell GTEx ranks number 2 in this and almost last in another one. It means that context mattered a lot when people were ranking, and we should think about that when we go for another round, for example.
And I think there's two kinds of context. One is the other projects,
inside,
but also how people frame them within. Like that same topic of GTEx was framed in three very different ways with different accompanying goals.
We framed
and so I think that just that diversity has some
interpret one way or another because the contexts were so different and the number of votes relatively low. So it's something to really bear in mind when you do interpretation later on. That's all.
Yeah, our goal was to actually integrate across all 4 topics and have a single one, and we didn't have time to do it. So think of this as like first pass, and then anything that ranked high in any of them would be considered in like a second round, and we may literally do another round of voting, if not here, then like remotely.
Rick Myers.
Rick Myers. It was bouncing, so maybe I missed this, but writing genomes, was that in any of these? I didn't. We talked about it.
Not in the end, actually.
It was embedded, I think, in one of the technology development items, I believe, but it wasn't its own thing. And you know, given the amount of airtime it got, I think here in a lot of what were generally positive comments, maybe that was an oversight, but that's a good point, yeah.
And is that Eric?
Yeah, and responding to this comment about disease, the genomics and disease, I think 2 things about that, that how this conference was organized is focused around variability. That could be normal variability or variability in disease. Plus, I think there's a common theme I'm seeing in everything that's hitting the top, is that people want depth and quality in order to do anything that they're going to do, whether it be basic biology or disease research. And I think that's going to be necessary to do disease-based research.
I just want to make one other observation about, I mean, I was looking in particular for topics, like the correlation between the amount of conversation we had about a topic and where it ranked, and were there any things that were discrepant. I think for the most part, feel free to disagree, but there was some correlation there. The one topic that maybe ranked higher than the amount of airtime that it got was training opportunities. So I just think that's worth highlighting, and maybe going and reading that one is something that clearly there was more interest in the topic, then we talked about it.
An important point. And in the brief conversation we had about that in this session, I was on the stage 4, we also talked about like the portfolio of like R01s versus big projects, and that's something that's not noted here, but yeah.
And Daniel, and then Eric.
One point I did want to make is around the complete atlas of genes and their functions, and this is one area I think where large-scale human sequencing will be key. It's not actually reflected in that card, but integrating the functional genomic approaches with direct assessment of variation that actually exists in the human population and integrating that together into this resource will be key. So it might be good to think about editing that later to make sure it does reflect the fact that there would be a human genetics component to that effort as opposed to it just being a purely functional genomics and perturbation exercise.
It's great to see all of it laid out. Yeah, I think in past NHGRI planning exercises, there comes a point where you want to ask for each of these things, what would it mean to do it? Is this a project that has some scale at which it actually delivers a well-defined comprehensive result? Is it an RFA for a bunch of R01s in a topic? Because I think it varies all over. Is it a directional thing for the Institute? And probably before it makes sense to, vote much further, it may make sense for you, like, what are they? Directionally, if you're saying, if the genie appeared, would you like one of these? That's good. But then nailing it down to which of these are communal activities that have a scale that would matter, and below that scale it wouldn't get there. And these are sort of things that have been helpful eventually for defining the larger projects versus the kind of direction, the kind of more open-ended technology directions. And it probably can't easily be done sitting here, but if NHGRI sort of digs in the next round to try to turn these into, actual projects and directions, that would be great.
Caroline?
So I mean, I actually think discussing some of that with this is important here. I want to say that we don't want you to get into the nitty-gritties of RFAs. There's lots of reasons why that's not a good thing to do. But I do think the sort of, is this a consortium effort? Is this a large resource? Or is this a well-served by investigator-initiated? But in that, to the other part, in some of these are very specific, and I can look at them and I can say, I've talked about this with other people. This is NHGRI's wheelhouse. I see what we're doing. This is some of these are larger, all of biology or wide things. And so also getting a little more specification, what is it that's to use Eric's phrase, the forefront of genomics in this? What is it that NHGRI could move the needle on in this that wouldn't happen if we weren't involved in it, or we could uniquely bring something to? As we're having that discussion, I think that's another important aspect of this.
Yeah, absolutely. Jay and I were trying to pull out some of the high-scoring things, and for each one, we thought we could have some discussion about the role of NHGRI in it. So he's going to put up some slides if we're ready, but okay, one slide. But quickly, is there anything else that was missed?
Yeah, I'd like to respond to what Jay said about the training opportunities. Actually, I think it actually did get ranked pretty high in topic 3.
No, that's what he's saying.
It got ranked high, but we haven't, compared to how much we talked about it. This exercise is useful to reveal how important it is to people. Yeah.
And to follow on what Mike Zodi said, are any real surprises?
I was surprised that epigenome didn't do better, but I mean, I think it's probably worth going over the top ones, which I think is what Jay is heading to do. No? Yes? Yep.
Yeah. So,
getting an adapter.
So this is just like, I was going to put up a slide for framing some of the questions, but
yeah.
Just go ahead with the first topic.
Okay, so why don't we go back to topic 1, which is discovery and interpretation.
Actually, I thought we were going to try to merge across.
You're going to merge across. Okay.
Yeah. So an attempt to identify themes that emerged across the 4 topics on the fly. I think we're going to try to. Jay took some notes, and he's going to bring up some that were high ranking. No. Here's your notes.
Look at
right
the
here.
There you go.
Okay. I will look at Jay's notes and bring up some that were important across topics. Okay, an easy one, I think. Tech dev scored highly, and it feels like it's quite in NHGRI's wheelhouse. Does anybody disagree with that or think that it needs refinement in terms of specific initiatives, data sets, knowledge, or capabilities, or is it pretty clear?
Well, this is Ross Hardison, Penn State. Yeah, the technology development's important, but there were, it seemed like there were multiple types of technology development that were captured in different topics here or breakout sessions before. So, yeah, I like the general thing, but I'd like to hear the specifics of what it is we voted on. And the text was really jumping around a lot. I couldn't read it very well.
Yeah. So I think long read sequencing came up in a lot of places, multimodal molecular profiling, technologies beyond bulk genomics. that look at
Large-scale
tissues.
DNA synthesis
Yeah.
is the other one, right?
Synthesis, yeah.
And I think, you know, it's a reasonable question. I don't think I have a strong opinion about whether these kinds of things, I mean, are there particular goals that would be best served by kind of, like, are they $1,000 genome type goals where we really go for it as like a goal in itself with a dedicated kind of effort? Or do we just want to move the ball forward on technology development in general with some prioritized areas? Like, are there technologies where the whole field, you know, where a meaningful advance would have a transformative impact? At least, you know, in my view, probably the one that stands out there is, I mean, some of these things are already, they're already moving along to some degree in academia or industry. You see new methods every week. Probably the one area where the community that develops these methods is much smaller and extremely good, but there hasn't been a broad-based effort in academia to try and really move the ball is the DNA synthesis area, right?
Well, and since telomere to telomere was one of the highest scoring, the technology you're going to need to do that, right?
And
through the centromere, through the repeats, right?
And so we'll certainly look at what was brought up in other breakout sessions that maybe not made it on here. I also know that Mike Smith is organizing some discussion about this. He had it last year at technology meeting, and I think planning it again. So this is not the last time we're going to be talking about broad technology development.
Adam?
Yeah, just a quick comment on technology development. I think there were some comments along the lines of not neglecting the possibility of new technologies to measure things we already think we can measure, because sometimes we learn a lot by the next alternative to RNA-seq or the next alternative to ChIP-seq. So I'd just like to have that on the record as something to think about.
So it's a really good point, and I think this has been raised in different ways here and there over the meeting. It's kind of an interesting thought exercise I was doing, like, you know, there's a reluctance of this community to engage with proteomics, I think, and this has come up again and again. I would imagine if we had a massively parallel protein sequencer that could give us fast Q files, right, that we would have a different attitude about it. And so it just kind of reflects almost a cultural dynamic with technology. And a great point. We're only profiling. It'd be good to focus on things that we can't do now at all, right? Or at least this community is not doing.
Karen,
but there's a lot of new approaches, you know, barcoding proteins and maybe developing new technology for protein sequencing may be one of the new technologies that the NHGRI may look for.
Yep, it's coming along.
Karen's had her hand up for a while.
Okay. Not to beat a dead horse to death here, which I feel like I'm doing, but I want to go ahead and emphasize that I think the telomere to telomere project is not something that should be tucked under tech development. I think we've reached a stage where we have adequate tech development that we need to think about this as being a specific initiative. This needs to be prime time. This needs to be organized.
Yeah, you got the top vote. I wasn't trying to tuck you under there. I was just trying to say, is there tech dev we need to do it, or it could help do it?
It was Sharon, Justin.
Yeah,
Aviva's gonna. You just turned off your mic.
Oh, it's not. Now it's on. There is, at least in the original phrasing, we tried to cram the text about that as well. It's just really hard to make something that doesn't take a whole page if you're, it starts reading like a research plan. But there were several items, and they are on the original slides as well. So if NHGRI wants to pull the tech dev that's needed for that stuff, then it's there.
after.
Sarah?
So following up on that telomere to telomere and that we already have existing technology, I would argue that we don't have existing technology to do this at a high scale, which is the main limitation for folks like myself. You have an individual lab, you have diverse samples. The sequencing technology has revolutionized things. Now we're doing hundreds of thousands of people, but we can't get through a few individuals for good reference genomes. So I'd like to see going beyond existing technology to get this so it's more cost-effective and can be done at high scale.
Can I comment? I think it was mentioned earlier by Eric that there could be a proposal on the table as to where existing technology is for doing this, how much tech dev is needed, and maybe a phased program beginning with reference genomes, et cetera, for different populations. And I think the group would be very interested at different levels in that, but I think we don't really understand, I mean, that was a very good comment, where the field is right now, what are the barriers to getting there. Certainly some can be done. Can it be done, 1, 10, 1,000, 10,000?
Adam.
Something that I heard that Elisa and others have talked about it a lot, in a number of these high priority areas is multi-omics and moving beyond DNA and RNA. And that's a traditional boundary for us, but a number came up, some proteomics, metabolomics, even imaging or tissue assays, and integrating all those different data types. So I just wanted to point that out and ask people generally about how strongly they feel that those multi-omics and beyond data really need to be integrated and what it takes to integrate them to get to those kinds of goals.
Hey, I just wanted to comment about the end-to-end assembly and the need for technology development. I mean, I think if you have, short reads and you're willing to tolerate if you're willing to tolerate 20,000 gaps, producing end-to-end assemblies is a solved problem now. I think the challenge is if you want to start to fill in those gaps, then you start to need to bring in long-read technologies. There's a lot more tech dev that's involved there. Those long-read-based assemblies, right now the state of play is that they reduce your single base pair accuracy pretty substantially, which can, which can affect, for instance, your gene annotations, introduce frame shifts inaccurately, et cetera. So that's where it gets complicated. So I think it's really the integration of the existing methods that can reliably do end-to-end genome assembly inexpensively from short reads with long-read technologies, with polishing, in order to be able to produce long end-to-end genomes that have very small numbers of gaps.
Right, so I think Adam's question gets into, computational resources. So should we spend a little more time on tech dev or then move on to what kind of computational tools do we need?
Why don't we, well, like within any one of these technologies, I don't think we want to get too much in the nitty gritty. I think, I do think we could either go to computational tools or to genome assemblies, which
yeah, no, I just meant
topic,
because Adam asked a question, but I think that question is going to take us away from tech dev. So I just wanted to figure out.
So why don't we do a few more comments on tech dev and then we can move on to something else. But.
I think we should move on soon.
Eric Mendenhall. Not in the nitty gritty, but I think with tech dev, we might have an opportunity to combine this with training opportunities. So some of the best tech dev comes from people scientists in their 20s, right? So using this as an opportunity to fund postdocs maybe for $100K for a year or two to develop tech dev, or young investigators, we could synergize these two opportunities.
Great.
Yeah.
Should we move on?
So why don't we? So computational resources, was that a good next topic?
Or computational tools was another high scoring, right?
Yeah, it scored highly. It was phrased in different ways. I just want to point out that some aspects of that were clearly computer science. Some of them were clearly informatics, databases, things like bringing algorithms to data. And then there were some things that were clearly statistical and kind of modeling aspects. It had a lot of parts to it. But those different parts all generally scored, and training and the workforce for comp bio. Right. So all of those generally scored pretty high. And let's open up some comments on how that needs to be refined and why NHGRI. Adam?
Yeah, one quick comment about computational tool development. I don't think we discussed it much in this meeting, but I think there is a distinction the community needs to be aware of between creative new algorithmic work and refinement, maintenance, documentation, software engineering work of tools that have already proven themselves to be widely used. So I'd like to have that on the table.
Yeah, I agree. That was part of the distinction. Within each of those categories, I think we need to make that distinction of innovation versus like the really important but less glamorous aspect of like dissemination and maintenance.
Right. So Adam asked the question of how important is integrating beyond RNA and DNA, and I would just argue from the medical perspective, it's like critical because RNA and DNA is little of what we have in the medical sphere. We have proteins, we have metabolites, and phenotypes. So I would just argue that being able to integrate across that is really critical. Ali?
Yeah, so I agree on that, and I would say that I feel like that's part of what's missing when we're trying to look at the data that we've collected, say in ENCODE and so on, trying to see what some of the downstream effects are. It's at the level of what's happening with a protein when you do a perturbation or something. I was also going to say that, I mean, the experimental method development and the computational method development go hand to hand, because as you generate a new assay, you're going to have to come up with new methods to analyze them. And as we continue generating more and more data, the biggest challenge is turning out to be how to integrate it all together. Ultimately, we've talked about it in this context that at some point we might get to the point where some of the hypotheses are going to be generated by the computational biologist that will lead to the next set of experiments that we might try to do. At that point, we're in a world of being driven by hypotheses again, as opposed to being driven by just let's collect 10 times more data and see what we can do with it. And I think that would be a healthy place to be 10 years from now.
Thanks. I really appreciate that comment. As someone who spent most of my career reacting to data sets and reacting to technologies, I would love to see the models driving what we're doing.
It's easy to generate data.
Yeah.
So.
Stephanie?
Stephanie Hicks, Johns Hopkins. I'll support that and say I'm very supportive of generating data from multi-omic, but not just omics, so non-molecular data as well, electronic healthcare records, imaging data, and then also those methods statistical methods and computational methods to integrate all that is non-trivial, and there needs to be support dedicated to computational people to to identify better ways of integrating that to effectively derive knowledge from that data.
Jason?
I was impressed to see how high the data visualization ranked, considering it was probably the cheapest of the topics on the list. And I'll also say that data analysis and computation statistics, machine learning are really critically important, but are kind of diffuse and ambiguous directions for the community as a whole, and have a lot of turnover, right? So that's more of a supporting the community. But the data visualization, when you develop good tools, they get used for a very long time, as we've all seen with the genome browsers that we have in hand. But I think it's time to really expand on that.
That's one of those topics I'd want to, again, I agree, like, that's something to consider, but it would be helpful to me and presumably others at NHGRI if we could spend a little bit of time, how would that be best organized, and what is it that really you see as NHGRI's role in the world of data visualization?
There are some amazing data sets already collected, right? That you could imagine more creativity and industry experience being applied to trying to take those, to visualize those data sets in new and unanticipated ways.
I mean, ENCODE came up multiple times, right? That it kept changing from 10,000 tracks to 30,000 tracks. I felt like over the since I'm not involved in ENCODE, over the length of the conference, the number of tracks just kept going up. But as an example of, you've invested all this money in generating the data set, investing some money in visualization might really lead to new discoveries.
Especially because, I think Jason's point that it's cheap is, in the grand scheme of things, especially given how much money has gone into generating these resources, it is a drop in the bucket to hire a couple of people for a year or two to knock this out. I don't know what the right mechanism is for that. Like, is it a challenge, or is it something else? I have no idea, but it does seem like a super effective, potentially effective use of resources to make more out of something we've already generated. Right?
And Daniel, you want to comment on it?
Yeah, just briefly, I think it's one area where it's key to have distributed resources rather than centralized. With visualization in particular, making sure you have lots of people who build, you have a centralized resource ideally with some kind of API, and then make sure there are lots of different groups who are trying different models for visualization rather than a single kind of centralized resource is absolutely key.
There's a lot of waving going on over there.
Marilyn Ritchie. Two things on to Katie's question about what should or shouldn't be in our wheelhouse around computational methods. I think we don't want to try to become NSF in terms of doing hardcore basic computer science, but I think engaging in the technologies and methodologies that are being developed in that space and bringing them into genomics is what we're lacking. We don't need to create another neural network. We need to figure out how can we take neural networks or support vector machines or boost learning and use that in the context of the genome in ways that make sense. Because computer scientists are grabbing our publicly available data and sometimes doing things that aren't very useful with the data because they don't necessarily know enough details of the data. So that's on the computer science I do think we need to invest there, but not in creating computer science. It's bringing that to our field. On the visualization, I would say if we do that on the cheap, we're going to end up with a 50-track browser again. Data visualization science, and especially if you do it well, which is having machine learning and artificial intelligence built in so that it's not just looking at a database and plotting things on a map. It's actually 3D, moving it around, being able to zoom in and out and go through the data, kind of the way video games are. So think about when you play a video game, how interactive and how the screens change. That's what we want with our data. We don't want just a map that we have to scroll and scroll down 30 or 50, or as we add more data, 100 tracks. So I think that is not going to be cheap. We need to engage in data visualization scientists and bring them to our data because we don't know how to do that.
Okay, great comments. So we just want to make sure we're moving through topics. So why don't we take one more comment? Nancy, you've had your hand up for a while. So
this is to Adam's question and to some of the comments Carolyn's made. Why NHGRI should take the lead on this is in part the history of demanding quality and insisting that the tools really fit the challenges and problems. And I think the other part to why NHGRI needs to take the lead, and there are a few other institutes that are broad enough to want to partner and maybe common fund initiatives. I'm not saying it all would all have to be NHGRI driven, but a key part of the integration, and it's been discussed, this transfer learning, the opportunity to move this into data where we primarily have DNA and RNA, the translation of all of this information from other things to data that we have a lot of already, the DNA and the RNA-based information, that's prime NHGRI territory. And that's why I think this is a topic that NHGRI, at the very least, would want to lead, even when other institutes must be engaged in the activities.
So why don't we just, in the interest of making sure that we get through all the topics in a timely fashion.
Aaron's one who brought up data visualization to begin with. I guess want to say one more thing.
I just wanted to let people know. So Anshul, Katie Pollard, and Carrie Getz and I have been talking about this throughout the meeting. We are trying to organize some sort of meeting to write some sort of white paper about data visualization, what NHGRI could do with it. If there's anyone who's interested in those topics, please just contact one of us. I think that would probably be helpful on this area.
Great, thanks.
So topically, I mean, why don't we next go into, I think, two of the, we can separate these out, but two of the topics that I think were represented on, each represented on more than one of the polls, and I think scored reasonably consistently highly on both. So one is this notion of functional atlases, and the other is next-gen QTL studies. And these are related in the sense that some of the phenotyping, molecular phenotyping methods that you might use as readouts are similar, but why don't we separate them and consider them apart from each other? And just to, as a divide, you can imagine the divide being things where we're we are introducing the perturbations, right, using synthetic methods, whether at the level of the element or the variant, versus things where we're taking advantage of standing variation, right? And so why don't we start with the first one?
And similarly, the environment can be controlled in the first case, and in the other, it's just whatever people have been exposed to. Yeah. So the first one, sort of the functional atlases in a experimental, controlling perturbation.
Yeah, so some of the axes here are like coding versus non-coding, variance versus, saturation mutagenesis or just scattered variants versus elements, and then, and what we would phenotype with Bing.
I want to make sure I understand what we are going to do. I think the functional atlas can be achieved through continual mapping of biochemical signatures such as chromatin accessibility marks, DNA methylation, and so on, but at increased resolution and in different biological conditions. It can also be achieved through the perturbation, I think, that you alluded to by massively perturb the genome at increasing resolution and using observations to inform us about their function. I think both are going to be needed for us to come up with a truly bona fide functional catalog. Either one alone may be insufficient or in danger of generating incomplete list.
So let me get that right, Bing. You are suggesting that the two topics that Jay mentioned, one was sort of the synthetic approach, targeted, versus the natural variation that could be used in these, so within a tissue, for example?
Sorry, if I understood the project well, I thought the project initially focused mostly on perturbing the genome and observing the consequence. I would like to emphasize the complementary aspect, which is continuing a lot of what we have begun with ENCODE, but now at single-cell resolution.
Yeah, so it's an important distinction to make, and I think, again, different manifestations of this topic either included or didn't include that first, that non-perturbative aspect of this. And so, yeah, I think that's actually a good point of discussion. So you can imagine this institute, in addition to, there is clearly a lot of enthusiasm for the perturbative approach, but in addition to that, should we also be undertaking the high-resolution characterization of biochemical marks at single-cell resolution in endogenous tissues of model organisms and/or humans, and how would that relate to other ongoing efforts to do this kind of thing, right? That would be important.
I would strongly argue that we do not forget doing that, because I think that's where we clearly have a gap in our knowledge base. Current catalogs that have been produced in the past 10 years are not at the resolution that would be, I think, of use in a single cell resolution, cell type resolution. So we have the technology, and we have the, I think, a lot of the know-hows and frameworks to do that. Why not we move this from a preliminary status to a comprehensive status?
But I don't think they're in conflict necessarily if you encompass individual variation with a number of, for example, people, where you survey those biochemical activities in a population, at a population level. Kelly had a question.
So one thing that had been brought up, especially in our breakout session, which I don't see up here, is the need to include different developmental stages and potentially the prenatal stage. And I think that the types of model systems which could really do this are the iPSC-derived cells, or tissue types, which really are primarily fetal-like. And the methods in developing those types of cell models are really advancing quite fast.
So I wanted to point out that in terms of this question of doing sort of just we hit every site in the genome and look at the effect, versus looking at, I don't know, natural populations, is the issue of epistasis. And I think, that's something to keep in mind. There is these complex interactions that are going to be very hard to a priori. I mean, I guess you could keep hitting every, 2 at a time, but it gets much more complex than that. So that's a reason that I think that's important to keep in mind. The other thing is I totally agree about this question of development, that some of these you perturb things, I don't know, in vitro, and how are you going to actually measure the phenotype? It may not be relevant depending on the stage of development in which it's expressed. And lastly, I may have missed it because I came a bit late, but I'm not hearing a lot about looking across sexes. I'm not hearing much about that. That's so important because we could see really different phenotypes between the sexes.
Right, and with regard to your first comment, I think one issue that hasn't been raised is doing some of these cell-based experiments across at least a common set of haplotypes, so that if, maybe not so much for loss of function, but certainly for missense. I mean, there's already lots of medical examples where missense alleles on different haplotypes have drastically different effects. So yes, you can't do every possible pair, but if you're going to attack a particular gene, attacking that gene on, I don't know, 6 common haplotypes across different populations might address a little bit of it in a practical way, but we definitely do need to keep that in mind.
John?
Yeah, I think it'll be important to define for NHGRI and to sort of propagate some standards, if you want to call it that, for what single-cell technologies are expected to bring to the table that aggregate technologies are not showing. I mean, it's like, think about NASA wants to build a new telescope. They don't just build it. They build the telescopes for very specific reasons. We're going to have this telescope because it's going to allow us to see X. And so I think defining what X is and what because clearly the aggregate data we can get a lot of value out of. Clearly the single cell data in a certain case, but in certain cases you can get a tremendous amount of value out of. But there's a massive gap in the genomic resolution of those. And so I think having some clear guidelines will help frame the application of these technologies.
I think Andy, you need...
So there have been several comments along the lines that NHGRI is going to be setting the standard for data quality, and we're going to get really good data, and it's all going to be wonderful. And the best way to assure that is to recognize that everything we do has error, and so that we need to quantify error throughout. And I think there should be explicit verbiage in this project in particular that we're going to think carefully about what are the levels of error at each step, how does that matter in the experimental design, and pull in statisticians to help with the design of these experiments.
Dave Valley, Hopkins. I think I want to follow on to what John said. The one thing that strikes me about the various perturbations that one can do and the single cell experiments that one can do, they seem almost unending. And so I think we have to really be clear about what the goals of the experiments are rather than doing these experiments just for the sake of doing the experiments.
So just if I can react to that. So I think it's important I think you're totally right. I think it's important to be clear about what the goal is, right? Is the goal to generate a bunch of data, or is the goal to understand the function of each element or gene, whatever that means? And I hope it's the latter. And if it is the latter, then you have to be very clear about definitions up front and about what particular kinds of progress will mean so that you can quantify success without moving the goalposts along the way, so to speak, right?
So there have been a lot of really interesting topics that have been brought up and voted on, but each one of them is relatively monolithic and addresses one particular element of the community. I think that it would be terrific to find a pilot project that actually brings across multiple of these themes together, and I think there is a very significant convergence, both in terms of technologies and methodologies and end goals that could actually make that happen. For instance, I think that with the advance in single-cell perturbation assays with full profiling, the saturation mutagenesis and a focus on a particular problem, for instance, studying how pluripotency is affected by variants. The entire community could come together and really go after these in a completely integrated fashion. And of course, that would require generation of the models that we have actually seen as one of the priorities and topic of computational tools. So I think, I completely agree with John, and I think this was one of the strong points that I also made yesterday, that rather than doing something in abstracts, it would be a lot better to do something once we know what particular problem we want to address. If as a community, you guys decided to choose a problem that is not in conflict with other institutes and bring all these various elements together, I think that would create a really valuable program, project, and pilot.
John? Ross?
So on this topic about perturbing, say, every nucleotide or every element in the genome, I'm assuming that this is done in the context where you're going to assay for what the impact is, what the phenotype is. And of course, we've had every day lots of discussion about the different levels at which you can determine phenotypes. And it's very complex. So I hope that this AIM will develop with some specific goals about what is the readout, and then I want to maybe inquire whether or not this idea of having a pilot project with a subset of the genome where you do many different levels and types of phenotypes and compare and learn from it, maybe even develop these transfer knowledge things from. Is that still on the table? Is that part of this, or is that gone?
So my I think it's very much on the table, and I think there was a lot of enthusiasm for it throughout, even though it maybe wasn't in here, and even my enthusiasm for it has gone from low to high over the course of it. And I would say also, just to speak to another comment, some of these things like epistasis, particularly for cis regulation, can be tackled if you're focused, right? So why don't we take, just one or two more, and then we can move on to the next topic. John?
Yeah, just following up on that, I mean, it seems one thing NHGRI can do that it's in a unique position to do and will cost $0 is to designate 1% of the genome, however you want to describe it, as the technology sandbox. Propagate that to the entire community. Inside NHGRI funded, everywhere. Everybody knows this is the 1% of the genome that everybody's working on when they develop any directed methods. And it's a reference, it's there. And again, NHGRI is uniquely, I think, positioned to establish that.
Given the amount of time that consortia spend recurrently trying to pick their 1% to focus on, I think that's probably a good idea.
Start now.
17Q21. No, okay. No.
Okay, should we move on to Eqtl studies on steroids, literally or figuratively?
Well, it's GTEx on steroids.
Or GTEx, sorry. Aviv.
I mostly want to make a technical comment on feasibility. I think these high-resolution studies, people often expect that you need fresh tissue for them, but I want to point out that that's no longer the case, and it hasn't been for a while now. You can isolate nuclei pretty much from every tissue. And it's accessible, and it works very well, and it even works on actual GTEx samples. So that's something important to bear in mind, just in terms of thinking through what needs to be done in a very pragmatic setting. The other reason that this is beneficial is that that allows multiplexing and can be done across individuals and within an individual. So you can easily accrue a lot of individuals and run in a very cost-effective way a large number of samples accrued over time. It's a different study design than, say, 3 years ago in that field, so it's important to bear in mind.
So are you saying that, I mean, particularly in pointing out the existing samples, is this like shovel-ready, so to speak?
I think it's less about whether you use the existing samples or not, because the existing samples have some constraints associated with them. And you might want to do things differently and so on. But it means that you can use the same collection pipelines that were used before, in which NIH invested a lot of money in setting them up. And moreover, that you can do a project that's really a big project.
I think this is a very good point.
Otherwise, it takes forever to be done, and it is unbelievably costly, but that's no longer the case.
This is a good point, because this brings up one of the things that's on the slide is, can we identify for NHGRI what's sort of production ready, what could be a project ready to go versus pilot versus a white paper, et cetera.
Yeah, and I think that Steve, I'm not sure if he's still here, was the one who brought up the warm autopsy. Part of that does come from the cancer experience where people are actually then making mouse xenografts with those live tumor cells. So that's the PDXs are a very important resource, which then gets used for a variety. So there are some reasons he may have used that particular terminology that relates to then actually taking living cells and creating PDXs.
It's an entirely fair point.
No, I'm just saying why it may have come up.
But I just wanted to highlight the design principle of how much you need to collect in a more specialized versus a less specialized setting. And also, we generally think about these things in postmortem, but I would emphasize that the examples that Howard had on his slide are accessible from living healthy humans. Skin, colonoscopies, there's a lot that can be done, and for which there's large collection efforts already at the scale that would allow you to do these kinds of mappings. It's a very realistic project today.
This is Ira Hall. I think there's a real opportunity here for the QTL mapping project to tie together a lot of the things that we talked about in previous sessions, and that we should think really carefully about that when we design it, if we design it. Some of the things we talked about in the last session about long-read sequencing and the dark matter of the genome, as well as somatic variation and epigenomic variation, can all be answered in the same sort of experimental paradigm if we do it right. And I'd also like to reiterate what Aviv said, which is that it would preferably be a pretty large study.
Tim Reddy, maybe this is obvious, but saving viable cell material, whether it's iPSCs or LCLs or something from these individuals would create, I think, a lot of long-term impact as well.
The current GTEx samples have LCLs and fibroblast cell lines.
Great. You want to go ahead?
No, go ahead.
Oh, I was just going to say there are a few that we have talked about, but before closing them out, maybe we should just touch base. Is there anything else on assembly?
So, well,
go ahead.
Let's make one point.
Jay has something on assembly.
I was kind of hoping to bring this up. So it is, there was some, I think, really nice breakdown within that one item that was quite popular about the different levels of this, right? So there's the telomere to telomere. Then there's, the points that Karen brought up, enabling genome biology, enabling association studies at the population level, and then also hitting, this is an opportunity maybe to try and get more genetic diversity or representation of human populations into our collective resources. So I just want to make the point that the goals are not dependent on one another, right? There are technologies today that are pretty darn good for getting, near complete assemblies that are scalable and could be used to attack some of everything except the telomere to telomere, and are much more sort of ready to go. And we shouldn't necessarily wait till we are able to do telomere to telomere genomes cheaply in order to try and work out some of those other things, or just start, right?
Yeah, and it relates to the somatic variation and mosaicism and the structural variants that have both been big topics in the meeting.
I worry this is something where, and this alludes to some of the points Eric was making earlier, where to get from where we are now to like 99.9% good will cost a lot less, by orders of magnitude than 100%. And as we're thinking about what we could do with scaled versions of this, we don't let the perfect be the enemy of the good. Eric.
Yeah, thank you for bringing that up again. And I want to say, sort of debunk some of the things said earlier in this session today, that actually I think we're closer to that goal than many people realize. And I think it's doable in the next 5 to 10 years.
Which goal?
Telomere to telomere, plus representation across the population. And that this issue about indels and so forth, or problems with base calling and long reads, is being rapidly fixed. And I have to say, with the study that with my colleague right here, Adam Philippi, we're working on for a vertebrate genomes project, which will include human genomes, we're actually learning what can be achievable now and actually give metrics to certain draft to complete genome assemblies that I think would be useful for this group.
Yeah, just to push back a little bit on this balance between rushing ahead to do some near-perfect genomes with what we've got versus developing new technologies. Of course, these very interesting regions are highly variable, and everything works against us. It's going to they're so variable. It's the rare variants in these classes of alleles that are going to be the very most interesting. So we need to be able to do a lot of them pretty well. So we need the scalability sooner rather than later, and that requires the technology development. So I urge we don't lose track of that.
The counter argument is you need to be able to impute a lot of them pretty well, right? And there may be some convergence there, right?
Yeah, I think we, day one, lost a little bit of hope in the idea we could impute some, and of course the very rare ones, that'll be even more true. So I still think the needle points a little towards technical development myself.
Andrew.
So just on the note, once we have these, telomere to telomere assemblies, the new regions that we're going to get out of it are, notoriously difficult, and all of the technologies we have to assess, or most of them, to assess regulatory regions and ChIP-seq, things like that, are short read, and those are going to be really difficult to map back to these new regions that, we're going to want to see what acting elements are there. So I think that's just an important point.
That's why Illumina bought PacBio.
Eric.
So the discussion really underscores, I think, the need to have clarity as to what these individual goals are. Because I think there's a lot of really exciting things on the table. I wonder, and I was talking to Adam a little bit about this, doing things like, say, on technology development, having a list of 10 great goals that we really feel are the most important things, defined with clarity, like how good does it have to be for the demonstration? I think there are a set of burning needs. Sometimes they're developing a new technology. Sometimes they're just getting data to a clean enough state. We talk about the ability to impute alleles down to a frequency of X in different populations. But we don't actually have on a website, here's really the target we would like to reach. Read DNA and RNA reliably in a cell in a certain way. Here's a really great target. The end-to-end telomeres, telomere chromosomes, is it a cost target, a quality target? I think creating a mechanism to debate and put up on an ongoing basis good challenge problems would help us a lot, because when we say we can do X, it's easy to say it and then say, yeah, but it's not really that accurate or that good, or it really costs this much. And it's not like these are magic, but sometimes declaring problems and defining them with crispness and clarity focus the attention of individual labs, they focus the attention of industry. Even in that Genome project, arguing what did accurate mean? How accurate was accurate? We eventually had to get down to a certain number of errors per 100,000 bases, which forced you to have a technology to prove that you were there. So I kind of think the discussion is wonderful for this morning, but it makes me crave a mechanism, some kind of either technology working group or data resource working group. When we talk about Eqtls, on steroids, the single cell steroid thing, to what level of accuracy do I want to be able to measure gene expression differences? I think all of that's going to drive us to a level of rigor and precision, and it's not going to happen without a mechanism to collectively define the challenge and then hold our feet to the fire. So I'm not speaking in favor or against any particular proposal so much as speaking for a mechanism to exist to identify these things, shine a spotlight on some of them, and make them clear enough to know whether or not we've actually killed them.
Yeah, no argument.
Amen.
Let's do it.
How about shifting gears a little bit, evolution, population? Oh, did you want to do something else?
That's where I was going to go.
Comparative genomics, evolution, and population genetics. There's been a lot of positive comments about that, and it's here a little bit in these, but I'd like to hear a little more discussion.
It's historically been the remit of this institute, right? And it did, it didn't get as much enthusiasm as one might have expected, and that could be, again, just the flavor of this particular crowd, but I think it's worth discussing, and I think it relates to these conversations about, how maybe it wouldn't cost that much, and maybe it would, relative to the size of the investment, maybe there are things that are worth doing.
But can we frame the discussion on the questions on the slide, not just broad discussion of
In which question on the slide? So what specific things, or why?
I would go with, if just any of those questions are fine, but not just like, why do we think, like,
what's the goal?
What's the goal and why NHGRI?
Yep.
And what's ready for prime time, or what's a 10-year vision?
Yeah, we've heard several times about comparative genomics, but not much about inherent variation within other species. And I think we've seen in primates some very dramatic data recently that show the value of that. So I'd hope the program includes systematic evaluation of genetic diversity within some key species and the weight you can use that to bring to human disease
I think
problems.
you're referring to the Kyle Farr paper from Illumina. Yeah, it's a worth reading in this context for sure. I agree.
Well, and our upcoming baboon paper, but this is not a time to trumpet that.
Yeah, Adam?
Yeah, I think at this point in time, the most exciting use of comparative and population genomic data is in combination with epigenomic and gene regulation data. And so some of that is in these targets, like with the sort of scaled up GTEx and with the idea of epigenomic data across species. But I think that theme of combination of epigenomic and evolutionary data is something to emphasize and something to sort of pair with any comparative and population genomic projects that are done at scale.
Ross is waving.
Oh, why don't we go this way then, Eric?
Oh, okay, so I would dearly love for you to add comparative epigenomics. And this is right along with what Adam just said, but just let me elaborate a little bit that as we pursue phenotyping these mutations in different model organisms, we really do need to know what their epigenomic profiles are. And I think there is the hope for some rules that will help us transfer the information more easily between species, even before you do the machine learning, and I hope the machine learning helps.
I mean, just along those lines too, maybe just to come back to something I think I had in the first session, maybe a different direction or a supplementary direction for the comparative angle here, which has always been a part of the mission, would be, single cell atlases for a broad range of related model organisms that would extend from not just comparative genomics, but a comparative description of the molecular phenotype of organisms that gives you insights into genomes and evolution, et cetera. I think that's where. Eric?
Yeah, I wanted to emphasize further on the comparative genomics. I saw that it didn't get such high votes as I wished, but nevertheless, someone brought up the topic that looking at gene variation, genome variation across species helps us understand what genes are controlling specific traits and the functions of those genes. And that is more easily obtained going across species than within one species. The other is doing comparative genomics within a species uses some of the same tools as doing comparative genomics across species. So within humans versus across species, I think will benefit in both cases. And then third, I would argue that there are some traits or some diseases that cannot be studied that easily in humans, like language, like we're doing now. And there are some species like parrots that have this ability to imitate like we do, but chimpanzees don't. And so, and we're finding that there are convergent changes on the same genes. And so you require a non-closely related species to study that trait. And I think we'll learn a lot more about that through comparative genomics as well.
Great. Let's wrap that up. We talked about that already. Yeah, we talked about modeling. Or do we want to talk about it more? I thought we had kind of talked about it.
Well, the only thing I would say is in population genetics, we didn't talk about human populations. And I do think we started out the meeting talking about diversity, but it didn't really make it all the way through, and maybe people feel enough sequencing is going on, but I think we continue to have some emphasis on population genetics with regard to the human population and better representation.
Just quickly echo what Ross just mentioned on comparative epigenomics. I think one thing that came up quite a bit in our discussion is epigenetic variation, which is very important. And I think lots of the topics that we ranked high actually get part of the epigenetic variation covered. But I think on the epigenetic variation across species will also help us quite a bit in understanding the relationship between a genotype, epigenotype, and a phenotype.
Great. Jay, where do you want to go next?
Oh, wait, so I mean, sorry. Go ahead, Karen.
I'd just like to comment on the idea of human population diversity as well. I think it was kind of a message that was weaved into a lot of the conversation but wasn't explicitly stated because of the enthusiasm perhaps on other calls with NHGRI. But I just want to re-emphasize that I think that most people I talk to are pretty understanding that the Human Genome Project is kind of obsolete in the fact that it only represents one person. So the movement and the enthusiasm to get these high quality genomes, as Jay referenced, and as I think people in the audience are estimating how good they should be, should absolutely be a priority if it's not explicitly stated here, and how to build the computational infrastructure around that, how it can be used, and the framework for wide distribution to kind of be the next Human Genome Project, for lack of a better way of saying. This kind of pan-genome effort, I think, needs to be on the top of everyone's conversation, and it's not.
Yeah, I just want to remark that actually in March we had a mini strategic meeting that outlined goals and priorities in that area, and that's already resulted in a concept clearance and notices of intent to publish FOAs to cover this area.
So why don't we go, Daniel and then Sarah, do you have a comment too? So Daniel, Sarah, and then is there another? Oh, sorry, Emma, and then we'll move on after those three.
Just expanding on the diversity question, I think there is clearly a push across institutes to get more diversity in, and I think we will see naturally more diversity, particularly in US populations, but there's not really a good effort underway to capture a global picture of human variation. We're still far from that, and I think there is still plenty of scope for a push on that from NHGRI. The other point is, particularly if we're interested in capturing things like, human loss-of-function individuals, I know you don't like knockouts, Sharon, so I'll refrain from saying that, then diverse bottlenecked populations will be key, and consanguineous populations will be key. And again, there's not current large-scale efforts underway in either of those areas. So I think it's worth explicitly pushing on that.
And those maybe have the advantage of being disease agnostic in a sense, those kinds of cohorts, which is maybe appealing to this institute in particular.
No question at all, that's right.
Sarah?
Yeah, I would second that, but just also say not just sequencing cohorts just to identify variation, but to have it very well integrated with phenotypic information. Right. And then also developing computational approaches, looking at things like signatures of natural selection that can kind of point into what are functionally important regions of the genome. So when you integrate that with then other functional genomics approaches, I think that will be really informative.
I mean, seconding obviously the phenotype and ability for consenting for recontact, but then also just emphasizing to get those selective signatures, we do need this at scale. So this is not just small scale efforts in each population, but really substantial numbers.
Emma? Why don't we go Emma?
Okay. So I wanted to, I think a lot of the things we've talked about are important, and Eric was talking about how do we move the field forward and help everybody's research move forward. And I just wanted to highlight, I think there's a danger of just doing like the incremental next step. So yes, single cell is an excellent technology. I think it's very exciting. But and then if we apply methods that we've developed more than 10 years ago, such as that we're going to do chip-seq across all of it and look what happens, there's a danger that we're not harnessing technology as powerfully as we can. And I think if we try and think about how we can move everybody's research forward with the most innovative approaches. I think Bing mentioned, like, a key thing is, yes, we can map where all of these proteins are binding, but a key thing is actually how can we do this with more in vivo in vivo information. So can we really measure binding in vivo, for example? And Adam was asking, okay, we've got all this sequencing data, reading and writing the genome, how do we integrate the protein information? But, and I think in terms of that, we don't just want to integrate what's happening now, we also want to think about how do we measure things in vivo. And I think in order to make the most progress, we need to think about how we can benefit everybody's research. And I'm wondering if we're going to try and integrate these ideas to get at those technologies more later. Or does that make sense?
Yeah, those are great points. I don't think I have an answer. But
okay.
Those are, so just in the interest of time, why don't we keep moving through topics? So we did touch on modeling, I think, in the context of some computational conversation. I don't know if we did it justice relative to the amount of weight that it got in the meeting as a whole. But just I'll bring it back up in the context of one thing we didn't really talk much about, but came up over and over again, and I think we only heard positive comments about, which is this idea of benchmark data sets and challenges, and how do you structure that and things like that. It seems like there is a lot of enthusiasm for that kind of idea. So do you have additional thoughts or comments about that?
Stephanie's got.
Stephanie Hicks, Johns Hopkins. I would completely and wholeheartedly recommend and support that. I find benchmark data sets are far and few in between good benchmark data sets. I can think of maybe one, for example, in single-cell RNA-seq, and that just came out and was funded by the Chan Zuckerberg Institute. And so I think NHGRI has a natural opportunity here to develop some really good gold standard data sets that a lot of people can use to develop computational methods and algorithms.
Trey, UCSD. Yeah, so I couldn't tell whether, I think there's two different arguments maybe that have been superimposed, but maybe you guys see what you think. I think some people are arguing the entire role of this institute is to create those kinds of benchmark data sets, and that's one of the major values historically of NHGRI. And so that's kind of the strong benchmark argument that underscores everything we do. I think there's sort of a separate thread on specific challenges like the DREAM or CASP challenges as a way of benchmarking, say, computational analysis techniques and maybe some data sets. So I don't know, maybe we can get some consensus on whether we're talking benchmarks as a huge value in and of itself of the whole institute, or we're talking about focused RFAs here.
So, to the first part of your question, I was really referring to the second thing, which is, data sets that could be used for the purpose of developing challenges for algorithmic development that would be broadly open, and how you would structure that, and also just what those would be. Right? Maybe that requires.
That is connected to the first comment. Sure. That really, if you structure, there I think are some people who have argued that that's essentially what NHGRI should be in the business of doing, is developing those kinds of deep, rigorous data sets.
Yeah, Adam.
That's a good question. I would argue, I wanted to weigh in on that question of Trey's and argue strongly for the second approach. So I just organized a workshop on standard simulation data sets for population genomics at Cold Spring Harbor a couple of months ago. And we had a huge amount of enthusiasm, but I thought the real value of it was that it came from a kind of grassroots community that really understood the issues. And I found that the methods developers, computational methods developers, really wanted these standardized data sets as well, because they want to be able to tell how they're doing. So I think there would be a tremendous demand for RFAs that could support the organization of grassroots groups that were developing benchmarking and potentially computational challenges or other types of challenges.
Yeah, and to the first point, I think this connects a little bit to the idea of like a part of the genome, the 1% that we include in all the assays, there was some talk about having some standards or experiments or candidate elements that were run in every experiment so that things could be compared more effectively or integrated and normalized more effectively across different labs. So I think I agree with Adam. I was thinking about it in the second way, but it connects to, if we're going to do the big atlases, can we do those in a way that they have these standards baked into them?
Jonathan.
Mike.
So a related issue is assessment of software. So as you know, there are often many different software packages that ostensibly do the same job, and they vary enormously in how well they work, how usable they are, and things like this. And so there can be tremendous value in forming groups that can systematically assess all of the different software competitors in a field and provide guidance to users.
And performance and usability don't always track together, which is an important
That's
point,
true
right?
too, yes.
And to Adam's point, it doesn't always have to be a competition. Like you get all those people together and maybe they'll learn like, oh, my thing's good at this, but someone else's thing is good at that, and maybe collectively come to a better solution rather than competitively.
I think there's a healthy way of framing these, and that's totally critical. So
Adam?
I just want a quick follow-up. I do think an RFA on these topics should be structured in a way that it makes sure something usable to the community comes out and not just a kind of a mealy-mouthed, everybody did pretty well kind of thing, which sometimes happens.
Sure, reminder, we're not supposed to talk about RFAs. Gabor.
I mean, I just want to point out how important this is and what a woeful lack of availability of these data sets for some of the very projects that everybody is excited about, like sequencing repetitive regions, calling variants in repetitive regions of the genome, or single-cell technologies that are coming online with basically no reference data sets available, or even some of the epigenetic bulk technologies, for example, methylation, DNA methylation sequencing. So full-heartedly support this and organizing working groups around topics that make sense.
So just to try and,
I would just point out, if we're going to talk about that, we need a strong component of discussing genetic privacy. I mean, a lot of the issues around why these data sets don't exist is because to optimally use them, you want them as easily publicly available to teams to play with the data, and that can be very limited. So I'm just saying, if we're going to do this kind of planning, there needs to be kind of a strong privacy component to how do you manage that.
And this goes just, connecting back to that. So we talked about warm autopsy protocols. So having reference individuals for which very comprehensive genomic, transcriptomic, proteomic, all kinds of single-cell bulk, all kinds of workup can be done. Maybe that would be one of the ways to go.
Folks,
Barak.
There.
Yeah. Barak.
Just address the issue that came up about the apparent conflict between black box machine learning models that are really powerful statistically, but maybe not so interpretable, and mechanistic models on the other hand, which I guess we all want, but are much harder unless you happen to guess right. So I guess there's probably an opportunity for intermediate models that help you move from more statistical black box-ish things towards more mechanistic things. I think there's probably new computer science in there. I think that's also might be the place where we were talking about models driving experiments. I think those intermediate models are probably places that will better suggest concrete experiments.
Yeah, so the point about involving the modeling people in the design is relevant not only to, I think, these sorts of challenges, but also more broadly across all of the experiments that we talked about, or projects. Marilyn?
To beef off of what Jonathan and Adam had said, I think in the topic 4, we also brought up the kind of platforms for these analysis tools and data sets. And I think moving toward an infrastructure and incentive to build these tools in some sort of shared cloud-based platform so that we don't end up with another whole series of tools that are not maintained and are hard to find and are hard to install. If we can kind of work toward a community where we're building things in the same space with the same data formats, I think that'll go a long way.
Great. Did you want to move on?
Yeah, so I mean, we probably are close to where we could wrap up. I mean, I think we've hit, I think, most of the major topics, if not all.
Should we just call that out?
Yeah, so I mean, what we could do is for a bit of time, I think, so one of the topics that we haven't brought up that was on the dot storm and scored, I think, somewhere in the middle related to biobanks. Some of the other topics that have come up include things like somatic variation, EHRs, PRSs. I'm sure there are other ones on the dot storm page or other things from the meeting in general. So why don't we just have a few minutes where we can have a bit of a free-for-all and
and also people who haven't spoken.
Yeah, especially people who haven't spoken.
Would like to hear from
Yep.
people who are maybe have an idea in their head but haven't been brave enough to speak yet. Gil?
So I want to make a general maybe suggestion that maybe can be of value in a future meeting. I enjoyed hearing the conversations over the past 2 days, and there's something of a smorgasbord approach that we're taking towards all of these topics together. I'm very curious what would happen if you actually had one discussion for technology development, completely separate discussion for data accumulation, and a third discussion for computational tool development. These are really apples and oranges to some degree. They require different expertise. There are different people who are good at each. If you just said, look, what is the most exciting technology development that we want to do? Rank those. Separately say, okay, we have some technologies where we can actually accumulate data now. Which ones do we think we want to accumulate more data in before others? And then you completely separately say, okay, what are the computational tools that excite us as a community most? Only computational tools, and rank those. And then try to, maybe in a fourth discussion, bring all these things together, because if we always discuss them as one, it's obviously always most exciting to do technology development, right? It's an empirical field, but your data accumulation is going to be less than optimal, and mostly I think your computational tools that will allow you to extract all the knowledge you could from the data you're accumulating are going to suffer the most. So I hope that would be a worthwhile point to consider.
Yeah, Adam Philippi, NHGRI. I'm co-chairing the Genomic Data Science Working Group. And so this meeting kind of springs out of the catch-all of general genomics. But as a data science working group, myself and others plan to have a future workshop in maybe the summer time frame that'll be entirely focused on tool development and methods development. And so these activities are going on in parallel to this. I just wanted to comment.
Just, I think maybe just to answer briefly, I think you want people from the other communities to comment on each other as well. So even in a forum like this, it would be nice to hear the other communities, no, I want a tool for this, I want a tool for that. So some kind of mixture.
David.
At the risk of making a point that's obvious, several of the projects that we've discussed this morning or in this discussion, telomere to telomere data acquisition and sharing, studying diverse populations, comparative genomics, all will synergize with the study of rare disease and complex trait disease, two funded areas that NHGRI is really promoting. So I think there's good sort of interaction and benefits across these goals.
Mike?
To expand on what my colleague across the aisle, Adam, said, that is part of and answer Gil's question, that is part of the logic of what we're doing. We're engaging within communities and in cross communities because we think they're both of value. So we're trying to get at both.
Lynn.
I'll just mention that because the issue of genomic privacy has come up several times in our discussion, the American Society of Human Genetics is preparing a perspectives piece on this issue, on genomic privacy. It'll be published sometime in the next month or two. We hope to hit the issue correctly, that is balancing the need for research open collaboration with protection of confidentiality. So if anyone has burning thoughts on that issue, genomic privacy, that you would like to see incorporated in our statement, you can contact me directly and I'll make sure that those thoughts are considered.
Okay. Okay, so why don't we wrap it up and turn things back over to Elise and Adam for some closing statements. I will just make a brief comment that I do think, one kind of consistent theme here has been or at least I think Eric nicely articulated, a lot of the amazing ideas that have been raised here, at some level, many of them are diffuse or phrased in different ways, and it would be a good possible next step to try and turn those into more clearly articulated, specific, ideally quantitative visions of where this could go, and then we could react to that. But.